about with a crossover, it makes a Holiday junction. The reason we're leaving it off is it's a little more advanced genetics than what we cover in Bio 311, and it's pretty hard to show without an animated three-dimensional animation. Here, we think about how Holiday junctions you have to have spend a little more time on it. Okay. So I'll do another like red means no and green means go. Here's do study. Okay. So in terms of as it relates to homologous recombination, there's a good figure in your textbook. Okay. In the 4th edition, this is figure introduction, part of that. And this time we have 2 chromosomes. So we have homologous, so we start with 2 chromosomes. Okay, one has mitosis or meiosis, right? You guys should know this. Mitosis metaphase. Yes, you would call it 5' end again happen. So they're proteins that aid or help promote what's called strand invasion. At this step, our red chromosome is going to be perturbed. Okay, it's going to have to change its type. Is the red strand has to be displaced. Okay, it has to move elsewhere. So that top strand on the helix on the red chromosome than what was intact. It's not the fact that the red chromosome is kind of bubbling outward. We're still have everyone's on. So as you're visualizing this in your head, back to its own blue strand. And ligase will finish that last connector. So let me get up here on the board what those resolved, it's what we call resolved DNA strands. So what this kind of our end pieces are going to look like. So we have we started when we moved back that braid, right? So kind of looking over here at step 2, what we started with. And a red chromosome. What I'm going to do to show you guys, so what you need to indicate, think about in your heads, however you want to indicate that on your notes. You could either use the color or some kind of wavy line. These fragments are not necessarily what the blue chromosomes originally started with, right? Because if we're in meiosis, they're copying off this red strand. So when we kind of shifting back to when we thought about mitosis versus meiosis, okay, the blue strands of DNA or the red strands could either be identical copies or they could be homologous, okay? Which means you might have different alleles on them. So when you come down and invade, and this blue strand comes down and invades and makes a copy of this red strand, it's potentially putting in different bases than what were originally there on that blue strand, right? So however you want to indicate that this has been copied from these two segments that are offset from each other a little bit, were copied from that red chromosome. Okay. So again, that could either be identical DNA to what it started with, or you could have a whole new set of nucleotides right now. Yeah.
I'm not sure about the reannealing. The book figure indicates that the blue doesn't go back to the blue strand, but the red, they kind of break up and recombine.
Yeah, the book has a blue fragment right here.
Yeah.
That's incorrect. Yeah, I hate that figure. So yeah, there's actually an incorrect, and it's funny because if you guys search online or if you have other genetics textbooks, you'll see it depicted both ways. And part of that is getting at whether or not you have two breaks or one break. And so it's where the diagram can be really confusing. So definitely come chat with the folks at ASAP, come chat with me if any part of it's confusing. What the level I want you guys to think about and understand is what I'm putting on the board. Okay. Yeah.
So you're saying that pink strand doesn't get any of the blue strand at all? It's just the blue strand's gonna get some of the red strand?
Yeah, not in this scenario I'm about to write. So this would be the way we showed it right here. This is a non-recombinant chromosome. Okay. So we fixed the double-stranded break, and there was no recombination, but you have copying because of the way the repair system works. You've copied a bit from the red chromosome. And this gets at both Sateli's question and Emily's question. If there is a crossover that occurs while this process is happening, you can end up with a slightly different mixture, and I'll write that here in a minute. Yeah. Potentially, yes. Yeah. Yes, because this pairing right here in this slide where it's slightly offset, and the hydrogen bond right here, these could potentially be not complementary to each other. And so you have to pull in other repair mechanisms to fix it. Again, though, the cell views this as a double-stranded break being much more detrimental than having a mismatch to go back.
The strands are not going to be identical in meiosis, meiosis.
Right.
But they are going to be identical in mitosis.
Correct. Yeah. So in mitosis, you guys, I mean, you could almost, and for mitosis, you could make these the same color, you know, and just track it differently in your mind, however that works.
So if they're identical, then they match up exactly where?
Yeah, they're identical as is the case in mitosis or in meiosis 2. Then, yeah, you wouldn't see any mismatches. This would all be the same exact recipe copy. Yeah. Everyone kind of with me up to this point, because this is where it gets a little hard again to visualize. But if these are recombinant, essentially this outcome, we went through steps 1, 2, 3, 4, and 5. Okay, the result, the final result is one of two options. You can either have a non-recombinant set of chromosomes, or, so this is not like a and thing, this is you either have a non-recombinant or you end up with a recombinant chromosomes. And this is if there's a crossover. So that means when we say crossover, DNA is broken and rejoined. And this would be part of the normal process. This break would be an induced break, not an additional break to the one that we started with that was due to some kind of damage. So this would be an induced break that resulted in a crossover. So DNA is broken and rejoined. And if this happens, this second scenario, then we end up with a different looking set of chromosomes. Okay? In this case, we're going to see red and blue on both of these fragments of DNA. So we have the same starting material in terms of our offset blue chromosome. Okay. And we have the red chromosome. But the idea here, I'm trying to line this up so it makes the most sense in your notes, is if you were to have a crossover occur, and you can imagine this anywhere on this chromosome. Okay, so this could be really variable. But where I'm drawing this dotted line, if you broke your DNA there and had a crossover and then rejoined it, hopefully you guys can kind of start to see that you're going to have all kinds of chunks and fragments of DNA. So this would be where, okay, we draw in our little red dotted line. So this, right now we look the same. Okay, I'm going to erase some stuff, so bear with me. So right now we look the same as a non-recombinant. Now, if you broke here, okay, and rejoined the pieces, that means you're literally taking this chunk and you're swapping it with this chunk on the right-hand side of the board. Or however you want to think about it, this whole chunk gets swapped with this whole chunk. Either way, you have a crossover happening right where that black line is. So that means down here, we're going to see this small fragment of the blue chromosome. Okay. And at the top chromosome, it's replaced with a red. So this is exactly how we've drawn it in the simplified version when we've drawn recombination before and crossovers, right? Where we had chunks of one color chromosome get swapped with the other. That piece is the same. The only thing that's weird is now you're thinking about where the origin of all this was because of that original break. Yeah, Jason?
So then, what causes the recombination to occur, or the non-recombination?
Other magical proteins. Yeah. No, it's there's other proteins that are in the vicinity that, and that's something that's been a big area of research because it turns out it's actually the bottom scenario is more likely to happen. You see more of the product, which is weird because normally we think of a recombinant being a more rare event. Whenever you guys have been counting progeny, right? You've been counting percent recombinants and thinking about that being the more rare class. But with a double-stranded break in the repair system, it turns out it's more often that we see a crossover event. Yeah.
And so is the DNA repaired first, and then it's all going on at the same time?
It's all happening kind of simultaneously. And the crossover, the breaking and rejoining happens while it's in this configuration. And those of you who are kind of curious, the rest of you tune out for 5 seconds. The holiday junction thing is when you actually have all this blue strand comes down in here too. So you have two strands that are crisscrossed with each other, and they're both breaking and rejoining, and the whole these X points travel either way down the chromosome as it's unzipping, and it's this whole crazy thing that happens. It's really cool, but yeah.
I think I'm asking.
Retune back in.
I think I'm asking the same questions he's asking. I just want to make sure that I understand it right. So it's not that you do this non-recombinant thing and then you get the artificial breaks and they switch. It's all kind of happening. It just so happens that it looks as if the top
yes.
portion is not intermediary to the bottom.
No, this isn't either or. These are two, so I'll write this on the board just so that two options for results. So like the end product of homologous recombination or repair, you can either end up with number 1 or number 2, what's happening up here or what's happening down here. Okay. And what Jason asked is somewhat along the lines of, well, how do we get option 1 over option 2? And that's kind of a whole separate monster that has to do with current research and stuff. Yeah.
Okay, to me, the way it looks like as you drew it on there,
Yeah.
it looks as if you go through one to get to two.
Oh, yeah.
Or,
I'll get rid of this guy then. You don't. Yeah. It's either you end up with this or you end up with this. What I wanted to show you was this is a direct result of what's happening in this figure, except for you break and rejoin. Yeah. And then when
Okay.
the piece is resolved, it either looks like this or like this.
I think I understand.
This is a really confusing topic. So if you feel lost and confused, you're normal. Okay, it's just hard to visualize this because as you guys know, this is all these different strands of DNA. So it's really weird to think about this happening three-dimensionally. So keep turning it over in your head and definitely come ask me questions, and we can try to draw it out in different ways as well.
Maybe here's another way to say it. So
Okay.
to me, it looks like to get to two, you have to go through one, and then another. You have is it two artificial breaks that you have?
Yes. Yeah. You have to break both. Yeah, both chromosomes have to be broken. Yeah. So crossover where DNA, when I say DNA is broken, both are broken. Does that last piece make sense to you guys that both chromosomes have to be broken for this number 2 option to